Good afternoon, everyone. Welcome to Psychology Grand Rounds this month. Today, we are delighted to have Dr. Beth Krone with us. Beth is a New York State licensed clinical psychologist and assistant professor of psychiatry at the Icahn School of Medicine at Mount Sinai. She is specialized in neurocognitive assessment, evidence-based diagnosis, and treatment of ADHD across the lifespan. And she has special interest in the intersection of immune, autoimmune processes, and neurocognition and executive functioning. Dr. Krone is on editorial boards of well-regarded peer-reviewed journals in the field. She's authored chapters in prominent medical textbooks and articles in peer-reviewed journals. She presents her research nationally and internationally. Dr. Krone has trained and worked in the Division of ADHD, Learning Disabilities and Related Disorders and the Newcorn Lab since 2008. She's currently the research manager and a researcher both there and in the Center of Excellence in OCD, Tics, and Tourette's. Dr. Krone is a board member of the Program for Protection of Human Subjects and a member of the Committee for Continuing Education for Psychologists at Mount Sinai. She's on the executive board of the American Professional Society for ADHD and Related Disorders and leads their communications committee. She's also a frequent contributor to the professional society blogs, and where she advocates for science-based education for practitioners. Dr. Krone is a consultant to several companies that are adapting evidence-based practices for digital technology and supervises doctoral students in evidence-based interventions. She supervises and provides case consultations for psychologists and graduate students and maintains a private practice. So in this 1 hour out of her week when she should be sleeping, she decided to do grand rounds with us. So Dr. Krone, welcome.
Thank you, Shilpa. I appreciate the introduction very much. I am going to share my slides here, and that should be shared now. Okay, so I am introducing the idea of innate and adaptive immunity and how they matter, how they're clinically relevant for ADHD. My conflict of interests, I don't really have anything financial conflict of interest wise, but my voluntary disclosure, I did some work with MindLift last year. They're a digital platform, but my main disclosure is personal. I am very interested in immunity and in autoimmunity. And so this is a new field that people have just started looking at in ADHD seriously. But I have gone through the literature going back, and I'm going to present that to you for quite a long time. There's a long clinical history of clinicians seeing an association. And the reason that I make this an upfront voluntary disclosure is we all know scientifically that correlation does not equal causation. And so my personal bias in seeing this, I just want to make sure that I'm very clear. I see correlations, but right now we don't have the causal mechanisms identified. And that's where I think research can play a role. So my objective of presenting the talk, why would I come to Grand Rounds and try to speak to everybody about this? I think that the idea of immunity and adaptive immunity and autoimmunity has really been highlighted over the course of the pandemic. Everybody sort of became, I guess we all became amateur immunologists just out of interest. And my interest in this is providing a translational overview of that research in context of ADHD. And I'm going to pull this through not just the literature, but some work that I'm publishing with some colleagues right now, taking this into the COVID arena, which is where we have some clinical relevance today that we can kind of put a finger on. So I'm going to provide a basic overview of immune processes and pathways that we believe intersect with ADHD pathophysiology, and describe some of those clinical risks at a population level. And the learning objectives to identify some of these immune pathways, understanding converging lines of evidence, and what those clinical implications really are. The clinical relevance for all of this is that there are various immune pathways, and they do likely contribute to phenotypical differences in presentation. And that can contribute to the way we view diagnosis, the way we provide psychoeducation for patients, and the immune liability, adding disease burden, needs to be viewed through an intersectional lens when we are creating our treatment plans. We inform our case conceptualizations by this, and we create interventions that target illness attributions, and ultimately it has an effect on people that I'll discuss so far as embodied shame and stigma for illness. I'm going to start off with something that all of you as psychologists know. ADHD is a polygenic neurobiological developmental disorder that's expressed in behavior and or cognitive abilities, and is often characterized by catecholamine dysregulation. What that means is that there are a lot of genes contributing to the presentation of ADHD in small increments. Some combination of genetic vulnerability and environment trigger the genetic changes and expression that result in brain-based manifestations of disorder. An individual's symptoms vary over the course of a lifespan, and often these symptoms cause at least some impairment throughout the lifespan. It causes impairment of functioning. We don't view ADHD as a superpower. We don't treat superpowers. We treat things that are hurting people. We treat impairment. It's quantifiable and visible and observable behavioral differences and cognitive differences that make people with ADHD stand out from peers of their same age and background. It is distinct problems with attention, working memory, response inhibition, executive control problems, and problems with producing, transmitting, and uptake of dopamine is a key feature of this in the research that we've done throughout the field. So these are the symptoms of ADHD per the DSM-5. The ICD-11 is coming into concordance with the DSM-5 with our 9 symptoms of inattention, 9 symptoms of hyperactivity, impulsivity, kids 16 and younger needing to have 6 or more symptoms, and people 17 and older having 5 or more symptoms for at least the past 6 months. An overview of innate and adaptive immunity. Innate immunity are the immune defenses that exist in our bodies and help us to fight against new pathogens that the body has not experienced before. And adaptive immunity are those immune defenses that are shaped by prior exposure to pathogens. A basic rundown of the components of the immune system for adaptive and innate immunity start with immune cells. It starts off with hemopoietic stem cells that are present in the blood, including cord blood and bone marrow. And they form these basic progenitor cells myeloid and lymphoid, myeloid in the bone marrow and lymphoid in the lymphatic system, that differentiate and produce the other cells that compose the innate and adaptive immune systems. B cells are an adaptive derivative of those lymph cells, meaning that these are, they start off in the bone marrow, but they are differentiated in the lymphatic system, in the spleen and lymph nodes. They produce immunoglobulins, which are antibodies. They present antigens that are found throughout the body to other immune cells, and they create cytokines that are released throughout the body. And we'll talk about cytokine mechanisms in a little bit. And they have a significant role in creating self-antigens, meaning that if you have an autoimmune disease, B cells can create the antibodies that are found in blood testing. There are natural killer cells that recognize the profile of your own body's cells. So we code our own immune systems from the major histocompatibility complex, which means that we inherit all of the mechanisms that we need for our immune response. We inherit that. It's genetically coded, handed down through families. On chromosome 6, it's a whole bunch of genes. It's polygenic. And it creates markers on our cell surfaces that respond to different pathogens. And when we create this signature, our natural killer cells can identify our cells and differentiate them from things that don't belong in our body. And it marks those cells for destruction. And when a cell becomes infected by a virus, or when there's something wrong with the cell, maybe it's cancer, or maybe there's another process going on, those natural killer cells will be able to identify those sick and broken cells and destroy them. T cells, pardon me, I'm sorry. Oh, this has to go down on the floor. T cells are another component of those lymphoid cells. They differentiate mainly in the thymus gland and then circulate throughout the lymphatic system. And there are a number of T cells that become activated and become shaped from the original T cell that is produced. There are killer T cells that remove damaged cell components. They are cytotoxic, meaning that they kill other cells and they produce inflammatory markers. There are helper T cells that help B cells to differentiate and do their job. And there are regulatory T cells that direct activity in the system. There are a number of others. If you're really an immunologist and you're really gonna study this stuff, there are gamma delta T cells and all kinds of other T cells. There's something like 25 different kinds of T cells. T cells used to be thought to only be in the peripheral immune system. We didn't originally think that they were in the central nervous system. But at any given time now, we know there are about 150,000 T cells circulating in a healthy person's central nervous system in the cerebrospinal fluid. We know that they cross the blood-brain barrier and they regulate the hippocampus and hippocampal neurogenesis. And we know that they can express different chemicals to promote neuron survival, and they play a role in neural development of the brain and maintenance of IQ. So T cells are pro-cognitive. Now for innate immunity, we have our common myeloid progenitors, which are primarily things that are developed within the bone marrow. There are two forms. There's granulocytes and agranulocytes. Granulocytes are cells that contain granules in the cytoplasm, and those granules contain materials that are toxic. They're pathogen to pathogens or to the cells around the granulocytes. When they release those little bombs of chemicals, it destroys bacteria and fungus, but it can also cause inflammation, just damaging tissues. Agranulocytes don't have those little packages within their cytoplasm. And there are various kinds of those. So what they tend to do, a lot of these are phagocytes, meaning that they envelop the pathogen, they envelop the bacteria, the virus, whatever it is that they're trying to kill, they envelop it and they destroy it. Neutrophils burst those granulocytes. They are the most common cell found in the blood, destroying bacteria and fungi. Eosinophils release those granulocytes and they kill parasites and larger cells and cancers. Eosinophils are involved in certain disorders like eosinophilic asthma, because those toxic products can also damage healthy tissues. Basophils release histamines. They're also involved in asthma as well as killing parasites. And they're prominent in anaphylactic allergic reactions. Mast cells also, they reside in connective tissue and mucous membranes, and they're responsible in part for permeability of vascular tissue. They release hormone mediators and cytokines, chemokines. They also release heparin and histamines. So they dilate blood vessels, they're pro-inflammatory, and they are they're important in mediating the blood-brain barrier. Monocytes, in vascular systems rather. Monocytes kill mycobacteria and fungus. They present antigens found in the body to T cells, and they are they include macrophages that are specific to individual tissues, different organs. They can enter the central nervous system, and when they do, they act like microglia in the brain. And they're dendritic cells, primarily outward-facing tissues like skin and the respiratory tract, the digestive tract, around blood vessels. They also collect antigens from outside and they bring them into the lymph nodes. In the skin, they're usually called Langerhans cells. And so we've got a lot going on in just within the blood and the tissues of the peripheral immune system. And just a little bit more about B cells. When they become activated by T cells, they produce immunoglobulins. And I promise that all of this vocabulary lesson actually goes someplace and has some relevance in mental health and with ADHD. I promise we're getting there. So these are different kinds of immunoglobulins, plasma antibodies, and complement. And how they interact with complement. So, IgE, skin and blood vessels, and sensitizes mast cells, contributes to allergies, kills parasites. IgG has 4 different kinds of IgG classes. One of them, opsonization, means that it's targeting different pathogens in the body and presenting them to be destroyed. Some of them neutralize different kinds of pathogens and shut those cells down. Some of them activate a complement system which tags pathogens and identifies them. It flags them for destruction. So different proteins, different peptides have different functions, and it all gets pretty complicated. Now, in the neural systems, we know that there is crosstalk with the peripheral system, with peripheral immunity. There are a variety of specialized cells. Microglia being one that we've mentioned and is mentioned a lot in literature. They are the main residents of the immune system in the brain. They do travel to the peripheral regions of the body. It's believed to be through injured tissue or across spinal boundaries. But recently, during the COVID era, when immunologists were looking very closely at the central nervous system, they found that microglia can actually just traverse the blood-brain barrier in small numbers. Radial glia occur along sensory pathways. They sense things like light. And they are responsible in part for balancing extracellular fluid balance. The oligodendrocytes have a role in immune homeostasis, and they are mainly found in white matter, and they play a role in myelinization, which is important when we talk about the neurological basis for a lot of mental health disorders. And we're talking about white matter integrity, and we're talking about decreased white matter or white matter hypointensities on MRIs. Oligodendrocytes are believed to regulate some of that. Astrocytes are some of the primary cells that are first created from the neural tubes, from those neural embryonic progenitor cells, and they signal to other glia. They help to actually create the architecture of the brain and provide structure. And so when we're talking about genetics and neural architecture for a neurobiological disorder, part of what we're talking about is how those astrocytes form and develop and how they move in embryogenesis to help create the brain and provide that structure for the developing brain. In the peripheral nervous system, Schwann cells are believed to act similar to oligodendrocytes, meaning that they myelate muscles, and satellite cells are in skeletal muscles, responsible for muscle development and regeneration. So there's that interface with muscle activity. And enteric cells are usually lining the outward facing, like intestinal epithelial barrier, and it communicates to the vagal nerve and through the vagal nerve to the central nervous system. So all of these are players in the communication between our brain and our bodies, and how we develop our brain and our bodies and contribute to our overall health and the health of our brain. These are the main pathways that these components work through: a neuropeptide pathway, a catecholaminergic pathway, which is interesting in context of ADHD, in that dopamine and norepinephrine and epinephrine play a role in this catecholaminergic pathway. There's a cytokine pathway, and there is the vagal spleen pathway, which is a cholinergic anti-inflammatory reflex pathway. And then there's adipose lectin pathway, which is being investigated now for its role in ADHD. That's an upcoming field. And then there's the blood-brain barrier direct route that we will talk about as well. So the neuropeptide pathway, there are very many more neuropeptides than I would be able to talk to you about. But I selected 7 with anti-inflammatory properties that are believed to have a role in psychiatric disorders, including ADHD. Cortico-releasing hormones signals these catecholamine pathways when people are under stress. And so these neuropeptides, which are small chains of amino acids that are produced by neurons, and they modulate neurotransmission in the central nervous system. They have action in the pituitary, and they have action in the vascular system, and they can depress cortical activity. They can affect learning and memory. They can affect hunger and growth regulation, and many of these are considered very important for immune system response. And so when we're sick, we don't feel very motivated or, having the ability to focus or pay attention, learning better when some of these things are regulated properly. And the immune system plays a role in regulating that. The catecholaminergic pathway was one of the first ones that was looked at in ADHD. Cortisol was identified in 1946, and by 1954, the allergic tension hypothesis of ADHD was developed. Spear said that ADHD was an allergic response to cholinergic dysregulation, because he saw that when people are having poor histamine responses, they tend to be sleepy and have difficulty focusing. And when they have too much adrenaline and norepinephrine, it puts that histamine response, that catecholaminergic pathway out of whack. Marshall in '89 revised that catecholamine pathway idea and brought in cholinergic imbalances. And so it became a pathway for stress or pathogens to act on the hypothalamus with glucocorticoids, and through the adrenal medulla axis on adrenaline, noradrenaline, and dopamine to affect behavior. In 2000, Ashton Jones added the locus coeruleus to this system. And that became the basis for a lot of research around how guanfacine works for people who have some types of ADHD. By 2019, Bus Kirschenbaum said that we needed to revise these ideas about catecholamines and cholinergic imbalances in ADHD. And so she put together a very nice review. And she hypothesized that an underactive HPA system caused dysregulated cortisol in ADHD, because in some people with ADHD, there is cortisol dysregulation, not that they have too much or too little, but that it acts differently than among people who don't have ADHD. The cytokine pathway is a newer concept in ADHD research. These small proteins, these interleukines, these lymphokines, these monokines, these are things that we've been hearing a fair amount when we talk about COVID lately. And we've been talking about cytokine storms. And when we talk about inflammation, and we're talking about systemic inflammation, these are the proteins and peptides and glycoproteins that we're talking about. And they act in different ways, regulating the immune system. And they have an important role in the body's response to different kinds of inflammatory prompts and different kinds of markers. And we'll talk about that a little bit more as well when we look a little closer at ADHD and some of the findings that we've got. The cholinergic inflammatory reflex, that's that vagal spleen response when people say that the gut is a second brain, and that the vagus nerve allows bidirectional communication between the brain and the GI tract. It's talking about this splenic pathway, how the vagal nerve downregulates the effects by triggering the cholinergic pathway. The cholinergic pathway being anti-inflammatory and stimulating those alpha-7 neural nicotinic acetylcholine receptors, because the acetylcholine is anti-inflammatory. And so it's that balance between the cholinergic and the catecholamine pathways. And this particular pathway probably intersects with the leptin adipose pathway, regulating insulin metabolism and metabolic syndrome, which is something that is being studied now in some groups that are working in ADHD and adipose and leptin. That adipose leptin pathway being a hormone, leptin being a hormone that's produced by adipose and some other organs, and signals the central nervous system by increasing neutrophil and macrophage activity and altering how B cells mature. Treg cells produce leptin and express high levels of leptin receptors. It has effects on innate immunity, increasing phagocyte activity, increasing cytokine secretion, and effects on adaptive immunity, increasing pro-inflammatory, should be pro-inflammatory IL-2 secretion from T cells. So in general, the adipoleptin pathway is inflammatory. And all of these pathways intersect with one another. And so the cholinergic inflammatory reflex, it triggers an anti-inflammatory pathway, where the adipoleptin pathway triggers an inflammatory pathway. And there are multiple ways that we know these different markers cross the blood-brain barrier. There are 5 of them that were written up in this Erickson article, talking about how neurovascular units interact with one another, how the barrier itself can be penetrated, how signaling can happen across the barrier if the barrier is not actually penetrated, and crosstalk back and forth. So that was a lot of immune stuff for a bunch of psychologists, but it has relevance to ADHD. I promised we'd get here. So there are multiple pathways in which it interacts, and we know that there are large genetic genome-wide association studies looking for the different places where some candidate genes for ADHD might influence neurodevelopment. Now, one thing that we know is these genes are pleiotropic, meaning that each of these genes not only has a role in brain development, but also has a role in immune functioning throughout the lifespan. A large GWAS found 12 independent loci associated with brain development on 11 chromosomes, representing at least 26 genes. And a lot of these have pleiotropy. So, for example, in that GWAS study, the highest association with ADHD was for a SPAG16 protein coding gene, which is also associated with chronic respiratory infections. Right now, GWAS studies are being put into giant meta-analyses along with functional genomic and behavioral data, and that's the new wave of research in large functional analyses, because it helps us to make sense of what how the genetics are actually manifesting. And so the functional genomic and behavioral and genetic data was recently looked at in fact, it's a 2020 article, it just came out, showing that for 11 psychiatric diagnoses, there are 152 independent loci. 20 of them are new, just found in this particular article. 9 have heterogeneic effects, and they don't have a specific marker that they can look at. But what the studies in general in ADHD tend to look at when they've been looking at the immune system are genes that predict how certain immune functions are going to happen. So looking at the way genes code for IL-16, and so they'll look at the gene that codes that protein. There were some studies looking at autoantibodies for DAT, for dopamine transporter. As a general rule, the studies that looked for immune markers were not done in a systemic sort of a way. It's heterogeneous populations, different ages in the lifespan, different races and comorbidities and sample types. And so there wasn't a specific pattern that we could find, because each one of the studies was really looking at something very different. And we have these gene marker studies, again, that are highlighting different findings in neurodevelopment. And they started off in about 1997 with gene markers looking at class 2 human leukocyte antigens that we found as coding for being associated with ADHD through its coding of a complement system in the immune system. And going all the way up through 2021 in GWAS, where we're finding these super pathways that are associated with immune response in ADHD. And the pleiotropy in neurodevelopment, what it can look like on a neurodevelopmental pathway, for example, with microglia are designed to accomplish brain development by pruning sections of the brain, pruning off neurons that we don't need. In the immune pathway, we know microglia will respond that way against viruses. They'll secrete major histocompatibility class 3 molecules, and they will prune, but depending on how a person's genetic code establishes these microglia, how they're programmed, they've been found to kill neurons or pathologically prune neurons, either pre or post-synaptically. And since we know that there's synaptic misregulation of neurotransmitters, this is potentially one mechanism that can account for that. Abnormal regulation of microglia can happen through a number of different contexts, and that can happen from a variety of different viral presenting viruses, and different viruses have different signatures for how they trigger microglia to improperly prune. And so depending on what the virus is, it can have a specific pattern of impairment that it creates. And also depending on when the person's immune system is triggered, depending on where they are in development, in infancy and even in childhood, and possibly in adulthood, it can affect the outcomes. One thing that we know, if ADHD is a disorder of dopamine dysregulation, and we know that a lot of these genes that are affecting dopamine transmission are also expressed on immune cells. All of our immune cells express dopamine receptors. And here's just a small chart that I put together. Dopamine is anti-inflammatory and it signals microglia. Dopamine activates naive T cells and makes them inflammatory. There are a variety of different cytokines and chemokines that are signaled by the different types of dopamine receptors. And so when it could be that it's not causative, but that it's linked in that the dopamine receptors that are not functioning well in ADHD are also not functioning well throughout the immune system. So where we come to now, and the reason that this came to my attention earlier, in 2017, there was a large population study in Denmark done by Nielsen. And in 2018, one was done in Norway by Hegvik, and in 2019, one was done in Sweden. And these were tens of thousands to millions of people in population studies. And they were finding that ADHD and autoimmune disease were highly associated with one another. Odds ratios were much higher for children to have ADHD in families where there was autoimmune disease. Going back into the literature, all the way to the beginning of the research, the first researcher looking at this in 1995 was a researcher named Warren, who found that ADHD was associated with those HLA genes for immune complement, and complement is related to autoimmune disease. His group then went on and did more research and found an odds ratio of 15.6 for people who had a particular immune-related signal in their genetics. And that was associated with autoimmune disease. In Middleman, Javier Castellanos was really interested in working with these groups to figure out what this immune component was. And his group was the only one in 1997, they did a cerebral spinal fluid assessment of ADHD, for cytokine markers, but they were looking at ADHD as a control group for OCD and schizophrenia. They didn't do spinal taps on healthy children for obvious reasons. And so they found elevations, but they weren't able to find consistent differences that made an argument in support of Warren and Odell's findings. Later on in 99, he wrote that he believed the issue was his sample was too small and too ethnically diverse to maintain Warren's findings. The one thing that all of these groups have in common is Warren, Odell, Middleman, all of them were looking at Northern European white participants from the same kind of homogeneous population that were Nielsen, Hedvig, and Lee as well. The whole group were looking specifically at one kind of population. And so I'm going to skip ahead now because I see that I'm just talking way too much. And so one of the issues that we talk about in ADHD is how do we treat it, right? And one of the questions of my research is looking at, will drugs for ADHD improve immune-related comorbidities or conflict with them? So for people who are susceptible and have things like Raynaud's disease and systemic sclerosis and lupus, where these things are prevalent, will treating ADHD be a problem for people who have these issues? And will comorbidities or features influence how we treat ADHD? So far as for COVID, we know that teens and young adults have a much higher odds of contracting COVID if they have ADHD. Untreated ADHD odd ratios are much higher. If they're medicated for ADHD, they do not have a higher risk. A lot of people, including myself, I'm sad to say, in the beginning, assumed that this was due to ADHD features themselves, that we were working with people who had difficulty paying close attention, washing their hands, keeping masks on. The kids were fidgety and hyperactive and couldn't stay in during lockdown. And we were attributing incorrectly, probably, that these behaviors were contributing to this higher rate of ADHD. We know that women and African Americans were at higher risk compared to men and compared to whites, with up to a 7 times greater risk of having infection. And we attributed that to lower socioeconomic status, greater amounts of stress, having jobs that paid less, so they had fewer resources, and a lot of social impairment. We did a study that we're publishing now with Lemut Health Services in Israel. It's the one that we're publishing now is the third in a series that people with ADHD not only had higher fevers and more often had pneumonia, they had greater severity of symptoms and were more likely to be hospitalized when they were exposed to ADHD. So it's not just having a greater risk for ADHD, but it's having a more severe course with worse outcome. We found that ADHD may be an actual risk factor for COVID, and that there may be a real biological, immunological vulnerability, making it not their fault. And so assuming that it's symptoms of ADHD that's causing the infection, actually it's got real implications for victim blaming and for shame and stigma associated with that disease burden. Helping these people to understand that it wasn't necessarily their fault is important, and it's an important part of the treatment plan and conceptualization. And I think that psychologists need to be aware of that when working with people that may be used to internalizing a whole lot of blame and shame for failures and assuming that it's their behavior that's causing it, when this is a real neurobiological developmental issue that may have real immune consequence as well. And that's my soapbox for the day. And now with long COVID, what we're publishing now is a paper showing that there are real links through probably the immune system for why people with ADHD are more likely to develop long COVID symptoms than people without ADHD. So if your patients did have COVID, even if it was asymptomatic, the UK Biobank has shown that even asymptomatic cases of COVID are associated with global reductions in brain volume and cortical gray matter thinning and loss of brain structure that is associated with cognitive and executive functions like an ADHD. So there's possible role for real pathophysiology there. And this is the paper that we are writing on this right now. So just to alert people that even kids who had non-symptomatic cases of COVID may have cognitive problems that are resembling or may actually be developmentally something like ADHD, and that people are going to need to really look at this going forward to provide services for these kids. I'll shut up now. Go ahead, Maria. I see you prompting me very politely. Thank you.
All right. Do you want to say anything in closing, Dr. Krone?
In closing, yeah, I just think that it's really important to recognize that these are real potential vulnerabilities and that there's something that we're going to have to face going forward, whether we're treating people for that stigma and burden, or if we're treating them for actual cognitive and executive functioning deficits. It looks like we're going to need more people in the field working on this.
Thank you so much for your talk, Dr. Krone. You can unshare your slides at this point, and anyone who's interested in asking a question for Dr. Krone, please put it into the Q&A section. If you'd like to ask your question aloud, you can raise your hand and we will have folks ask questions after the written ones are answered. You do have one question right now, Dr. Krone, in the chat. So here it is. It's from Veronica Taricos. Sorry if I'm mispronouncing your name. I'm interested to know links between ADHD and children on the spectrum. Do you have any response for that?
So ADHD and children on the spectrum, it's very common comorbidity. It's something that is clinically seen a lot. If you're asking about pathophysiology of the link between ADHD and spectrum, honestly, I'm not an expert on that. But there are definite immune pathways involved in spectrum. So yeah, I'm not the best person to ask about that in particular. But there's reason to think that there should be some commonalities there because it happens so often.
Thank you, Dr. Krone. We do have a hand raised from Julia Erhan Sangenito, and I am going to allow you to talk now. One moment. Go ahead, Julia. You should be able to be heard.
Yeah, I think so. Okay, great. Thank you for taking my question. I was just curious about the HPA axis and the cortisol levels, and if there was any particular subtyping. So for example, if a person has a diagnosis of ADHD and trauma, would they be the category that would be more likely to have that HPA axis disruption? Did they look at it in terms of comorbidities and changes in the axis functioning?
That is an excellent question, and it is something that there is not any consistent definitive answer on. I know that a lot of people look because kids with ADHD are often exposed to more trauma. Having ADHD can predispose you to being in a traumatic environment. A lot of kids with ADHD come from more chaotic home environments. So there is a lot of trauma within the ADHD population. But there's no definitive answer on that in the literature at this moment. It's something that I know people are looking at, including some of the people on our team.
It would be interesting to see the results. Thank you.
Thank you for that question. Just going to move you back into that other section. Hopefully that will work. Okay, Dr. Krone, there's a question in the Q&A from Stacy Lurie. Two questions, actually. The first one is this. Do the data suggest that children with multiple autoimmune diseases, such as T1D and celiac, are more likely to have ADHD?
So there is some research on celiac and ADHD because there is a relatively high prevalence of ADHD among people with celiac disease. Clinically, from a clinical perspective, we tell people to get their celiac as under control as possible before treating the ADHD, but there is a known association in the literature.
Okay, and the second question there is there a definitive pathway or pleiotropy for T1D and ADHD?
I don't think anybody has looked at that.
Thank you for that.
Be interesting to look at it.
It would be. Anyone else who has any questions, please go ahead and type them in or raise your hand, and we still have some time for questions. I'm curious, Dr. Krone, now that you've done some of this research, are you interested in pursuing this further? Are you interested in exploring this further in your professional day-to-day kind of research life?
Is that your question?
That's me.
You know me, I'm interested in everything. But yes, actually, I find this very, very interesting. And at this point, there's enough data. There are big data sets, and Mount Sinai is making things available all the time. New data archives. So there might be some stuff that can be looked at that way.
Thank you. Well, we'll just give another minute or so for any questions.
Okay. See, I could have kept talking.
And just as a reminder for everyone who's logged on to here, and thank you for registering. After the talk, you will receive a CE certificate link. You'll have to fill out a survey, and you'll be able to get your CE certificate that way. We have a new question for you, Dr. Krone, from Lisa Cohen. Are there any interesting avenues with regard to anti-inflammatories for ADHD?
So I've only come across one or two articles that were beginning to suggest that from a research perspective, in OCD right now, about 30% of the new drug applications are immune regulators or anti-inflammatory in some way. So I don't think that it's really being explored yet. I don't think they have concrete mechanisms at this point that they're translating, but I know that more studies are including immune markers in their in what they're collecting in their labs. So I think that it's an area that's evolving, and it may be fruitful in the long run. But right now, I don't think they've got anything.
Thank you for that.
Mm.
And we'll just, oh, there's one more, Dr. Krone, from Stacy Lurie. When choosing ADHD medications, for example, stimulant versus non-stimulant, are psychiatrists beginning to take comorbid autoimmune disorders and these immune pathways into consideration?
I think that if anybody is, that psychiatrists would be. I think that they're very much aware of the effects of stimulants and other ADHD medications. And I would believe that anyone who's experienced in treating ADHD should know what the range of the medicines they have available to them can do. That said, not every training program in the world recognizes the importance of treating ADHD. I think maybe I'm accustomed to who we have here at Mount Sinai, and they're just very good at what they do.
Thank you for that, Dr. Krone.
Mm-hmm.
I think we do need to wrap up this talk. It's 12:59. So thank you everyone for joining us. Thank you, Dr. Krone, for an excellent presentation. Our next grand rounds, just as a reminder, is going to be in June, on June 10th, 12 to 1. And as we mentioned earlier, you should receive in your email after this way to obtain your CE certificate for today. There's also going to be a recording available of this talk if you're interested in that after the talk itself. Thanks again, everyone, and have a good weekend.
Thank you.